time for discussion. I will call on our speakers here for some closing comments here in just a quick moment, but I did want to just, while I had the floor, to thank again Raj Koshi for his work on hepatitis over the years, his work as a colleague while I was at CDC and now here at the Coalition, being a great partner at NIAID, and I wish him the best in his retirement and look forward to staying in touch with him. I just wanted to see if, we're really all about developing these functional cures as well as maximizing the opportunities that we have with our current treatments. I think Jeannie really set us off well by showing the big burden of hepatitis B and what we're trying to reduce, recognizing it's the leading cause of liver cancer, the primary liver cancer, the third leading cause of cancer deaths around the world, really leading into our discussion. So I had a few. Just wanted to pose some questions to the group quickly, beginning with Anna. You really think there's a real enthusiasm for expanding treatment with our current treatments, but you laid out well sort of the just sort of the limitations of those therapies. They have real benefits, but then they also have some real limitations. We talk a lot about simplifying treatment, but it seems like you also laid out a real need to find ways of simplifying monitoring. How frequent do you think people with chronic hepatitis B need to be treated, need to be monitored to capture those that are, changing their natural history so that treatment would be warranted, number one? And number two, do you think we should have more age-based recommendations to start treatment, given some of the reasons you raised in your talk?
So monitoring, obviously, the more frequent, the better. On the other hand, we also have to be practical because this is not about monitoring for a year or two. You're talking about monitoring for years and decades. And I think the first couple of years is the most important because you really want to establish whether this patient has active disease, quiescent disease, or in between. Once we have established that the patient is in the inactive phase, you can do monitoring once a year. And I think that once a year is about as infrequent as it should be, because otherwise you would miss those opportunities to start them on treatment. In terms of how liberal do we make the guidelines, if you have Hep C treatment, something similar to Hep C treatment, it's just a no-brainer, right? You get a cure, oral treatment, 8 to 12 weeks, very safe. It's just limited by cost. But whenever you're talking about many years of treatment, it's not just that you start the patient on treatment. If you don't have the patient's commitment to stay on treatment and stay with the program, you're kidding yourself that you're doing the patient good. You give them a prescription and you say bye, and you don't have the mechanism of following them. And that's where I sort of have some concerns about, let's just treat everyone, because we don't have the capability of monitoring everyone. And obviously, the more educated patients are more likely to be adherent than others.
Thank you. Thank you very much. Let me turn it over to Stefan. He can have some remarks, Stefan, just from the other presentations. But you raised your efforts to develop a point-of-care test for Delta infection, and what are your prospects for that? Is it an antibody test or a virologic point-of-care test? What are you looking to develop, please?
Mm-hmm. Yeah, thanks for the question. I think the under-testing of Delta is not only a problem in, for example, African countries, it's also a problem in the US or in some developed countries. Yeah, and we're really lacking information on epidemiology, and we have to identify by a cheap method Delta, probably best together with HBs antigen. So we've developed a pan-genotypic test. And we are in the process now of getting that to the market, which will take a little bit time. It's a blood test for self-testing in principle. And we hope that we will have that soon. And I think it's very important, not for retract testing or reflex testing, for example. You can, of course, use that as well, but for really testing in field, yeah. And and we will see. And of course, we will keep you updated for the testing. And the final goal is then also to combine it with S antigen so that you have antibodies, IgGs against Delta and S antigen in one test. That would be a good first test. Of course, has to then to be evaluated whether Delta RNA is there in order to treat patients if there's available drugs until that time.
Excellent. Thank you. Look forward to see how that development progresses. Let me turn it over to Adam. Just invite any closing comments you'd like to make, but just in terms of capsid assembly modulators and sort of getting over to Chari's remark about the sort of the universe of companies and individuals working on new therapies. What's your view of the prospects of developing therapies that will actually be available for patient use? Is that still a long-term target, or you think it's one that could be realized in the next 3 to 5 years?
I wish I had a crystal ball. There's 3 separate questions you're asking all at once. The economics of companies working on HBV and the good fortune to come up with the right hypothesis and then executing on that hypothesis. A problem is there are fewer and fewer, as Chari points out, there are fewer and fewer companies working in the area and willing to invest in the area. Companies seem to be fairly gun shy, and similarly, study section panels tend to be very conservative. We need people and companies willing to take on high-risk, high-return projects. 3 to 5 years. There are some advances in recent history with the core protein directed molecules that have me incredibly excited. The apoptotic effect of some molecules, as a for example. But unless we execute and invest in the preclinical work, we'll never bring it to the clinic.
That's right. Thank you. I sort of have the same question for Tao and for Barbara. Sort of you're in the area of therapeutics you were touching on. What's your crystal ball as far as the drug development leading to patient care with new therapies?
I think the immunologists already have the crystal ball if they understand how the acute hepatitis B is cured, right? Yeah, I think the using pharmacological intervention with cccDNA by small molecule remains very challenging. Everything is preclinical now, and even just in the lab is under cooking. So cccDNA is really not a very easy target because it's not a single molecule. It's a big complex, and most likely a lot of the the antivirus targeting cccDNA will be the host targeting. So there's a lot of issues, but I do think if we put more effort on that and do a really large scale high throughput screening for cccDNA, we will find something that can open new doors, like Adam's company called Door, right? We can open new doors to looking for cccDNA inhibitors. And also those information will be greatly helpful to understand the cccDNA biology and further develop targets for targeting the cccDNA.
That's great. I was really interested in your HBX interaction. I think that's really sort of a new area as well.
I think
Barbara, what are your
yeah.
thoughts, please? Oh, sorry.
You
Sorry,
know,
go ahead.
I again would like to make the point that I think we cannot expect to restore the immune response in chronic patients to the level that those that spontaneously recovered had, because in the chronic patients, they never had such an immune response. The priming, the generation of the immune response in the neonatal or early life stage is completely different. So the easiest thing would be to have sterilizing immunity or to have antivirals that completely eliminate cccDNA. Otherwise, we need to find something that maintains an immune response in chronic infection to a level that controls. So if you transfer the bone marrow, that works. But otherwise, how do you maintain an immune response to that level so it continuously controls cccDNA? That's, I think, difficult. So I would definitely, crystal ball means we need antivirals too, and we need a combination of everything.
Thank you. Very good. And John-Min Hu, you started us off with an overview of the replicative cycle, et cetera. So after hearing these talks, do you have any other thoughts you'd like to share?
I think I would just like to concur with Barbara has just mentioned. I think we do need everything. In fact, I doubt with the nukes alone we can ever achieve 100% inhibition of even just replication, let alone the pre-existing cccDNA. Okay, so if we could have combination of inhibitors, that maybe we can have a deeper suppression of replication, which hopefully can turn into at least a blocking of new cccDNA production. Maybe let the host, the cell, take care of the pre-existing cccDNA, or the immunologists can figure out a way to target the infected cells and somehow get rid of the infected cells. And beyond that, I think I agree we need more research and more efforts. Thank you.
Thank you. Well, one of the byproducts, I have to say, of these webinars is to keep hepatitis B related research in front of NIH, to let them know that people are interested in this area of research, and just to help raise awareness and the education level of what has been achieved to date. It's great to hear from Jeannie her interest in hepatitis B, and I think that's a very good sign for us to continue to see how we can expand this research portfolio in the future, and hopefully, working with other institutes like NIAID, of course, as well in that regard. So I think I'll close it off from now.